Right now, I work at a company that makes the medical devices, which I will talk about it in my later slides. Most of my talk is pertaining to my doctoral and postdoctoral studies at the NIH. Giving you a brief introduction about my talk, I will be talking more about epithelial cell migration, and as you all might be aware and know that the epithelial cells in the cornea migrate as a sheet, and the migration starts along the basal layer, along with the basal stem cells at the limbus when there is a wound or an injury. And one of the important proteins which is involved in cell-to-cell adhesion is CDK5, which is a protein which is involved in neurodegeneration. And I will be talking to you about that and its application in corneal wound healing. What are the other proteins or markers that are involved in cell-to-cell adhesion or specific to the adult epithelial stem cells? This is something the future has to tell us, and there is limited knowledge and literature. This is a slide which shows the epithelial stem cell migration. On the first panel shows you the limbal tissue. The pigmented nature is because of the sample from the Indian population or from the cadaver source. And you would see a small little migration of tightly packed cells, and some of these are the migrating pigmented epithelial stem cells in the limbus. These cultures were pulse labeled with BRDU and chased for a period of 3 weeks. So the slide on the right, you will see that this is an explant, and after 21 days of culture, the epithelial cells are migrating as a sheet. The green labeled cells are the BRDU labeled retaining cells or the stem cells. These are present in the basal layer, and the ones that you see on the suprabasal layers are the transient amplifying cells, which are the daughter cells of the stem cells. Some of the cells below this dotted line or the Bowman's layer is actually the fibroblasts or the stromal cells that were activated during injury, and they also retain the label after 3 weeks. But what is important here is that the migration at the leading edge is in the form of a sheet. So the cells remain intact, the cell-to-cell junctions, adhesions are intact, and they still migrate as a sheet. These are cultures or the outgrowths which was in fact used for transplantation onto human subjects who had stem cell deficiencies or disorders. These are cultures showing small, tightly packed cells, labeled, retained, and these are the stem cells. And you see these phase contrast images on the right that show that these cells are very tiny, unlike the large cells on the rare corner, which are sparse and might have the junctions in between these two cells might be quite different from the tight junctions that are present in these population of small, tiny cells. This is another image showing the stem cells that are intact on the explant after 21 days, and these are pigmented as you see here, and these are the label retaining stem cells on the limbal explant. Similarly, we had isolated the cells or the tissue from the donor, an autologous patient whose buccal epithelium was isolated, and we cultured the cells. And you see these are the holoclone colonies, which have distinctly packed smaller cells, indicating that these are holoclones. And when they were transplanted, and after 6 months of post-op, vision was regained with these patients. Now shifting gears a little bit and trying to learn about the cell-to-cell adhesion junctions. This is a typical diagram showing the extracellular portion of the E-cadherin complex, or this is a junction that shows the extracellular domain and the cytosolic domain. The E-cadherin or N-cadherin is the protein of interest that I would be talking to you about right now, because this is one of the most important protein that is expressed in the corneal epithelial cell-to-cell adhesions. And CDK5, as I spoke to you all earlier, is a protein which is important in the neurodegeneration but has been less studied in cornea. But our lab at the NEI was interested not only in the lens epithelial cells, but to understand if CDK5 plays an important role in the corneal epithelial cell-to-cell junctions. So some of the other proteins at the cytosolic domain that I would be talking to you about is the IQ gap, the Rho kinase, which is the G proteins, and the RAC kinases. So when the cell migrates, the manner in which the Rho and the RAC actually behaves is different. So you would see in my later slides what happens to the activity of Rho and RAC when these junctions come apart, or when these junctions are together intact, how the Rho and RAC and the IQ gap proteins behave. This is a diagram showing the CDK5, which is a cyclin-dependent kinase 5, a serine-threonine kinase. On the right, you see this is the P35, which is the active form of CDK5. When P35 is bound to CDK5, it activates the Y15 portion of this molecule, and the phosphorylation of Y15 is known to activate CDK5. So an active CDK5 is also can be called as a Y15. These are human corneal epithelial cell lines which were stained for CDK5 and E-cadherin. So this slide shows us that both the CDK5 and the cell junction protein E-cadherin co-localize. The similar results were seen with the immunoblots, which were co-immune precipitated together. E-cadherin and CDK5 co-localized together, and as demonstrated in the blot on the right-hand side. In order to study the cell-to-cell adhesions, we created a cell line deficient of CDK5. A stable cell line was created, and as you see in the left-hand side, CDK5 staining was in green, and in the knockout cell line, the SH-CDK5 was devoid of expression for the most part. Of almost 90% suppression was seen, as you see in the Western blot on the right-hand side. For some reason, this was a simple experiment that we had carried out. This was done to show that the cell-to-cell junction formation was dependent on E-cadherin and not N-cadherin. So two different cell types were chosen. One is the HCLE, which is the normal corneal epithelial cells devoid of CDK5 using a pharmacological inhibitor called as olomoucine, which not only inhibits the kinase activity but also the expression. And another cell line which I had made in the laboratory, which was the SH-CDK5, which did not express the kinase. And the other two were the lens epithelial cells, which express the N-cadherin. So as you see in the bottom two panels, in the presence of calcium, the lens epithelial cells formed larger junctions. In fact, the cluster of cells in the presence of olomoucine, that is, in the absence of CDK5, the lens epithelial cells form larger junctions. The clusters were larger. In contrast to the second panel on the right, where you see that in the presence of olomoucine, a pharmacological inhibitor, or in the absence of CDK5, the clusters or the junction formation was smaller. So this slide suggests that the junction formation was dependent on the type of cadherin that the cells expressed. So it was E-cadherin and not N-cadherin. I'll go to the next slide. So the graph represents the same. The Sh CDK5 cells show reduced clusters, or the cell-to-cell junctions come apart in the absence of CDK5, and therefore these cells are dependent on E-cadherin, unlike the N-cadherin expressing lens epithelial cells. In order to further confirm this concept, we used a cancer cell line called as the MDA-MB231 cells, which did not have any expression for E-cadherin or N-cadherin. So these cells were transfected with GFP expressing E-cadherin and GFP expressing N-cadherin. And in the presence of olomoucine, you would see that these cells expressing E-cadherin had reduced cell-to-cell adhesion, or the junction formation was reduced in the presence of olomoucine, whereas the same pattern did not occur in the N-cadherin containing cells. So this confirmed that E-cadherin is required for cell-to-cell junction formation, and CDK5 is also required for stable cell-to-cell adhesions. In these cells, we also tried to look at the activity of Rho kinase, which are Rho and RAC kinase, which are small G proteins. And as you see here, in the absence of CDK5, the Rho activity goes up. And there is an inverse relation to the RAC. RAC activity is reduced and Rho activity is increased. In literature, it says that increase in Rho activity is associated with cell migration because the cell cytoskeletal proteins change. The alpha actinate in the cells is reduced, and the other proteins like the IQ gap will go up, making the Rho activity as well go up. So that's what we see in this slide here. In order to confirm that Rho activity was dependent on CDK5, we used a pharmacological inhibitor for Rho kinase. And as you see here, that in the presence of C3 transferase, which is a Rho inhibitor, the clusters or the junction formation increased. But when there is two inhibitors, both C3 transferase and olomoucine, as you see in the last panel, the size of the clusters will reduce. And that's what you see here in the graph, indicating that it is the effect of olomoucine that reduces the cell-to-cell adhesions. And in such a condition, the other cytoskeletal protein, another gap protein called the GAP1, increases. This also is suggestive that in the cells devoid of CDK5, when IQ GAP1 goes up, the cells come apart. The junctions become loosened up and they come apart. Or indirectly, CDK5 is required for stabilizing the IQ gap containing cell-to-cell junctions. This is a slide that shows E-cadherin, and P120 is another catenin which is involved in cell-to-cell adhesion. We stain these cells, the normal HCLE cells, and these cells devoid of CDK5. And as you see in the Sh CDK5 cells, there are pigmented particles of E-cadherin that you see which is internalized in the cytosol. That pattern was not visible in the P120. We also estimated the the border to internalized fluorescence intensity, and we see that there was a reduction in the Sh CDK5 cells. Since there was internalization, the border to cytoplasm fluorescence intensity was reduced in the Sh CDK5 cells when compared to the HCLE cells. We also confirmed this fact by surface-biotinylation, and we showed that since there is internalization and degradation at the surface in the Sh CDK5 cells or in the or in the olomoucine treated cells, the E-cadherin expression goes down by surface-biotinylation. This is another technique called as the total internal reflection microscopy that was used to study the E-cadherin junctions as well as the P120 catenin. Each of these particles or vesicles containing these cell-to-cell junction proteins were arbitrarily labeled, and the velocity with which these particles moved from in and out of the cells were measured using turf microscopy. All these red lines indicate the manner in which these particles moved over a period of time, and the measurements were recorded. This is for P120 catenin. You see that the green is not as long enough as you saw in the previous slide, because the migration or the movement of P120 from inside to the outside of the cell was not long enough, or the P120 was not internalized. It remained on the cell membrane, both in case of the normal cells or in the absence of CDK5. This graph shows that the amount or the rate of internalization was more in the presence of CDK5 inhibitor and as well as in the ASH CDK5, whereas P120 had no effect. This slide shows that since P120 remained at the cell borders and only E-cadherin was internalized, we tried to look at the N-cadherin expression. And a co-immune precipitation of N-cadherin and P120 showed us that the N-cadherin expression increased, suggesting that there is an epithelial to mesenchymal transition. So to summarize, in the presence of CDK5, the cell-to-cell junctions are stabilized. In the absence of CDK5, the cells dissociate. There is dissociation of P120 and E-cadherin complex, and E-cadherin goes through a lysosomal pathway. It gets endocytosed, and whereas the P120 containing vesicles are now expressing N-cadherin at the cell border, and they have gone through a transformation. Therefore, CDK5 is required for a stable cell-to-cell adhesion. Well, to tell you in brief about the company that I work for, this is a medical device company which uses plasma technology. Plasma is the fourth state of atmosphere. And this is the company that is located in Tacoma Park, Maryland. It is used for organ, we make plasma scalpels and devices which is used for various cancer surgeries, and it's used for coagulation at the same time. Since this plasma technology is known to selectively destroy the cancer cells, it is interesting to study the way in which it would affect the cell-to-cell migration and selectively apoptosing the cancer cells. This is the medical device that the company manufactures in-house. This is the future of plasma research that we would be carrying out. And this is Dr. Jerome Kennedy, who is the CEO of the company. And thank you for your patience.
Thank you very much. We're a little bit pressed for time because I guess we went a little over. If you have questions, please talk to Dr. Parthasarathy directly.